Hello, welcome to World of Microbes. Today we are gonna learn about immunology, its history, types and different defense barriers in details. For thousands of years mankind has been intrigued with the causes of disease and the concept of immunity. The prehistoric view was that disease was caused by supernatural forces, and that illness was a form of theurgic punishment for bad deeds, or evil thoughts visited upon the soul by the gods, or by one's enemies. The adjective immune comes from the Latin word immunis, which means exempt from public service. If you're protected, or exempt, from disease, injury, work, insults, or accusations, then you're immune. The first written descriptions of the concept of immunity may have been made by the Athenian Thucydides in 430 BC, described that when the plague hit Athens, describing about those who recovered from plague can nurse the sick, because they would not contact disease for the second time. The first clinical description of immunity, which arose from a specific disease-causing organism, is probably a treatise on smallpox and measles, Kitab fi al-Jadari wal Hasba, which was translated in 1848, was written by the Islamic physician al-Razi in the 9th century. In the treatise, who describes the clinical presentation of smallpox and measles and goes on to indicate that exposure to these specific agents confers lasting immunity, although he does not use this term. Until the 19th century, the miasma theory was also widely accepted. The theory viewed diseases such as cholera as being caused by a miasma, a noxious form of bad air. If someone was exposed to the miasma in a swamp, in evening air, or breathing air in a sickroom or hospital ward, they could catch a disease. Since the 19th century, communicable diseases came to be viewed as being caused by germs or microbes. Variolation is the intentional inoculation of an individual with smallpox material which was traced back to 16th century in China. Variolation used a lancet or needle to introduce pulverized dried smallpox scabs or pustule fluid into the skin of an individual. This resulted in a milder form with a much lower fatality rate, that is 2% to 3% than natural smallpox infection. However, a person contracting smallpox through variolation could still transmit it to other individuals, and variolation spread other diseases, such as syphilis and hepatitis. Variolation was Africa, India, and China, and in the absence of a better solution, variolation was adopted in Europe and its colonies in the 1700s. Edward Jenner is known as the father of immunology. He is known as the father of immunology because he was the first ever scientist to create the smallpox vaccine, which was probably the world's first vaccine. The terms vaccine and vaccination are derived from varioli vaccini, which means pustules of the cow, devised by Jenner to denote cowpox. He used it in 1798 in the title of his inquiry into the varioli vaccini known as the cowpox, in which he described the protective effect of cowpox against smallpox. In Jenner's time, smallpox killed around 10% of the global population, with the numbers 20% in towns and cities where infection spread more easily. Jenner postulated that the pus in the blisters that affected individuals affected by cowpox, which is a disease similar to smallpox, but much less virulent, protected them from smallpox. On the 14th of May, 1796, Jenner by inoculating an 8-year-old boy, who was the son of Jenner's gardener. He scraped pus from cowpox blisters on the hands of a milkmaid who had caught cowpox from a cow. Jenner inoculated the boy in both arms that day. The boy had a fever and some uneasiness, but no full-blown infection. Louis Pasteur was a French chemist, pharmacist, and microbiologist renowned for his discoveries of the principles of vaccination, microbial fermentation, and pasteurization, the last of which was named after him. His research in chemistry led to remarkable breakthroughs in the understanding of the causes and preventions of diseases and the foundations of hygiene, public health, and much of modern medicine. He is regarded as one of the founders of modern bacteriology, and has been honored as the father of bacteriology. He was studying bacterium causes foul cholera. He isolated the organism and induced cholera when injected into chicken. After some time, when old culture of cholera was injected in chicken, chicken survived. When freshly isolated pure culture was injected in chicken, chicken suffered from cholera. When same fresh culture of cholera was injected in previously inoculated chicken, chicken survived, remained healthy. In 1890, E. von Behring and S. Kitasato described the mechanism of immunity. Behring received Nobel Prize on 1901. They demonstrated that serum from previously immunized to diphtheria could transfer the immune state to unimmunized animals. He concluded that has antitoxin, precipitin, bacteriolysin, and agglutinin activities. This gave the concept of humoral immunity. Mechnikov in 1883 described that cells also contribute to immune state of an animal. He observed that some white blood cells were able to engulf microorganisms. He named it as phagocytes and observed that they are more active rather than antibodies. Later this leads to the concept of cell-mediated immunity. In medicine, the immune system's way of protecting the body against an infectious disease is called immunity. The main jobs of the body's immune system include fighting disease-causing germs, that is pathogens like bacteria, viruses, parasites or fungi, and to remove them from the body, recognizing and new substances from the environment, and fighting disease-causing changes in the body, such as cancer cells. There are two types of immunity: innate immunity and acquired immunity. Innate, or nonspecific, immunity is the defense system with which you were born. It protects you against. Innate immunity involves barriers that keep harmful materials from entering your body. These barriers form the first line of defense in the immune response. Acquired immunity is a type of immunity that develops when a person's immune system responds to a foreign substance or microorganism, or the person receives antibodies from another source. The two types of acquired immunity are adaptive and passive. In adaptive immunity, the body's immune system responds to a microorganism or vaccine, producing antibodies that can prevent future infections. Adaptive immunity can last a person's life. In passive immunity, the body receives antibodies from an external source, such as maternal milk or immunoglobulin infusions, instead of producing them itself. Passive immunity provides immediate protection, but only lasts a few weeks or months. The innate immune system constitutes the first line of host defense during infection. It plays a crucial role in early recognition and triggering of a pro-inflammatory response to invading pathogens. The innate immune response is mediated primarily by antigen-presenting cells, APCs, such as granulocytes, macrophages, dendritic cells, DCS, cytotoxic NK cells, T lymphocytes. They are regarded as relatively nonspecific. Now we will talk about the defensive barriers of innate immunity. Number 1 are physical and anatomical barriers, which are the body's first line of defense against pathogens, and include skin. The most important mechanical barrier, the skin is a tough, intact barrier that prevents microbes from entering the mucous membranes. These membranes line the nose, mouth, lungs, and urinary and digestive tracts. They are made up of epithelial cells that are bound by tight junctions, bony encasements. These provide a physical barrier to prevent microbes from entering the body. Endothelium. This is a single layer of endothelial cells that line the inner surface of blood vessels. It controls the exchange of substances between the blood and tissues. Next are physiological barriers such as intact skin, temperature, vigorous mucociliary clearance mechanisms, low stomach and bacteriolytic lysozyme in tears, saliva and other secretions. Endocytic and phagocytic barriers include ingestion of extracellular macromolecules and particles respectively. Lastly, barrier created by inflammatory response, tissue damage caused by wound or invasion of microorganism in sequence of events called inflammation. Here is the list of different organs and tissues along with the innate mechanisms protecting skin or epithelium. The skin contains antimicrobial peptides that play a key role in the skin's defense against pathogens. AMPs have many functions in the skin, including fighting pathogens as they are effective against bacteria, fungi, parasites, and enveloped viruses. Supporting the immune response as they stimulate cell migration, proliferation, and differentiation. They also regulate the production of cytokines and chemokines. Promoting wound healing and maintaining the skin barrier. Sebum is an oily substance produced by microscopic glands in hair follicles that protects the skin from drying out. It also lubricates hair and acts as an antibacterial and antifungal agent. Next is mouth and upper alimentary canal, which contains enzymes and antimicrobial peptides. They have directional flow of fluid towards the stomach, which maintains low pH. The acidic environment of the stomach, bladder, and kidneys and the bile of the intestines inactivate many viruses and bacteria. Various body tissues are protected by the low pH environment created by acid production. Contains digestive enzymes and the flow of fluid go towards intestine, which have normal flora, digestive enzymes, antimicrobial peptides, and lastly the feces expelled from rectum. Proteins and peptides play a critical role in the immune system, and are involved in both innate and adaptive immunity. Tears contain lysozyme that destroys the peptidoglycan layer in the bacterial cell wall causing its lysis. The continuous washing of the eye with tears containing lysozyme generally prevents the growth in the eye. Lactoferrin is widely represented in various secretory fluids, such as milk, saliva, tears, and nasal secretions binds to iron, thus interferes with the acquisition of iron by bacteria. Secretory leukocyte protease inhibitor, SLPI, is an evolutionarily tropic protein expressed at mucosal surfaces, mainly by epithelial cells. S100 proteins are a group of small proteins that play a key role in the immune system by regulating a variety of functions, including immune cell activation, inflammation, nutrient depletion, tissue remodeling, etc. Epithelial cells and cells of the innate immune system produce small proteins called defensins. There are many types with various antimicrobial and antifungal properties. However, the main two types found in vertebrates are alpha and beta defensins. Beta defensins help epithelial surfaces to colonization. They can kill microbes by creating channels and disrupting the microbial cell membrane. Alpha defensins are found in macrophages, neutrophils, and intestinal Paneth cells. Cathelicidin play a role in innate immunity by acting as endogenous antibiotics and modifying the response. Surfactant proteins are collagen-containing lectins that bind to pathogens, allergens, and other particles, enhancing their clearance from the body. They also modulate the inflammatory response and regulate the functions of other immune cells. Endocytosis is the process in which the macromolecules contained within the extracellular tissue fluid are internalized by cells. This forms small endocytic vesicles. It occurs through two processes, pinocytosis and receptor-mediated endocytosis. In pinocytosis, molecules are internalized through nonspecific membrane invagination. Whereas in receptor-mediated endocytosis, macromolecules are selectively internalized after binding to specific membrane receptors. Endocytic vesicles, also known as endosome, are acidic in nature, which dissociates. The dissociated molecule contained within the endosome are routed along a different pathway and fuses with primary lysosome. Primary lysosome derived from Golgi complex and contain proteases, nucleases, lipases, and other hydrolytic enzymes. Primary lysosome form as secondary lysosomes. In this, macromolecules are digested into small products like peptides, nucleotides and sugars. Later it eliminates from the cell. Phagocytosis is a type of endocytosis that involves the ingestion of large particles, such as microorganisms and dead cells, via large endocytic vesicles called phagosomes. Phagocytosis is found in many types of cells and it is, in consequence, an essential process for tissue homeostasis. Activation of phagocytosis can also occur indirectly by the phagocyte recognizing soluble proteins, called opsonins, that have bound to the microbe's surface, thus enhancing phagocytosis. This process is called opsonization. Once opsonins are bound to the surface of the microbe, they are opsonin receptors on the phagocyte, activating phagocytosis. Phagocytes recognize the target particle. Phagocytes signal to activate the internalization machinery. The cell membrane engulfs the target, forming a phagosome. The phagosome fuses with lysosomes and endosomes, a highly acidic compartment. The target is killed and digested by lysosomal enzymes, and lastly digestion products release from the cell. Barriers created by inflammatory responses are: Number 1 is tumor, in which swelling is caused by increased vascular permeability, accumulation of fluid, that is edema, and extravasation of leukocytes into the area. Second one is rubor, which is redness caused by increased blood volume, also vasodilation, and platelet leaking into the area. Next is chala, that is heat caused by increased blood volume. And the last one is dola, which is pain. Acute phase inflammatory response is a local inflammatory response, which is accompanied by a systemic response, tissue damage. This response is marked by the induction of fever, increased synthesis of adrenocorticotropic hormone, increased production of leukocyte, production of a large number of proteins. All are produced by the liver, called acute phase proteins. Now we will talk about the cell types of innate immunity. First one is neutrophil, which is first line of defense, first cell type that migrates from the blood to the site of infection. It is essential for innate immunity against bacteria and fungi. Its antimicrobial activity takes place by phagocyte rect, or by opsonization. Next one is macrophages, which are activated by TLR binding to their ligand, due to which phagocytosis, respiratory burst increases. It also secretes cytokines Illinois 1, Illinois 6, and TNF-alpha, as well as produce complement protein, and express higher levels of MHC class II and thereby present antigens to T cells. Next are natural killer cells. They are critical first line of defense against viral infections. It can distinguish between an infected and UN-infected host cell. By killing the virally infected, eliminate source of additional virus. These cells secrete cytokines IFN-gamma and TNF-alpha. These cytokines stimulate maturation of dendritic cells. IFN-gamma mediates macrophage activation and regulates Th cell development, whereas dendritic cells are critical from innate to adaptive immunity.